and we have worked on the de novo design of transmembrane pore-forming peptides, which had applications against antibiotic-resistant bacteria by killing it. So let's give it a bit introduction of the transmembrane pore. So these are the bag-like structures with the hydrophobic side facing the peptide hydrophobic side facing the hydrophobic lipid tails, and the hydrophilic side of the peptides facing the pore lumen. And because it is having a centralized channel or the pore, it can allow the passage of organic molecules which are bigger than water and ions. So this kind of peptides have several applications in medicine and biotechnology, and one of the important medical applications are the antimicrobial peptides, because by forming pores, it can directly kill bacteria by leakage of the bacterial cell materials. Similarly, it can also kill cancer cell lines, so that's why it has applications in cancers, anti-cancer agents. And of course, because of the rigidity and the stability and the compactness of the barrel pores, it also has applications for the biotechnological, in the biotechnology field, like the single molecule sensing and sequencing. But so far, there are a few examples for this transmembrane pore-forming peptides in nature, and one of them is the alanine peptide, which is an antimicrobial peptide, and it has been extensively studied for the antimicrobial activity and also the ion channel activities. So because of this lack of natural resources, people also have studied or redesigned such peptides from the natural channel containing proteins. But the problem with this kind of proteins are like it is very difficult to understand the role of each and every amino acids in the peptide sequences or the protein sequences to understand or to introduce the point mutations for specific purposes. So that's why people have started also doing de novo designs, because of the available structural features for the folding of alpha helix and also for the self-assembly of these peptides. But at the same time, de novo design is very complex because there are several complex interactions involving peptides, lipid, and water and ions. So that's why there are a few successful de novo designs so far, and these are the ion channels and the double barrel pores. But the ion channels are very narrow, so they do not allow the passage of bigger molecules, and at the same time, the double barrel pore, which are made of helical hairpins or helix to helix structures, are large and complex to introduce the point mutations. So, and the most important information so far which is missing are the generalized design principles for this kind of peptides. And in absence of which, it is very difficult to design such peptides from scratch, because the peptide sequence to function relationship remains mostly unclear. And at the same time, the role of individual residues in the previous examples remains mostly unknown or barrel specific, the interactions. So that's why it is very difficult to introduce the mutations for specific purposes of these peptides. So to understand the role of each and every amino acids in the peptide sequence for the stabilization of transmembrane pore structures, we performed molecular dynamic simulations. We started with an optometric peptide pore, because these pores are the smallest nanopores to allow the passage of organic molecules which are bigger than water and ions. And it is known that this peptide needs to be amphimatic in nature, which means that the hydrophobic side is in one patch and the hydrophilic side is another patch. And we chose leucine and isoleucine for the hydrophilic patch because of its high hydrophobicity, and similarly glutamine and asparagine on the hydrophilic side because of its high polarity. At the same time, another important criteria are the length of the peptides, and we chose 30 amino acids so that the helix can span the thickness of the membrane, and the 22 amino acids on hydrophobic patch so that it can span the hydrophobic thickness or hydrophobic membrane core. And we focused on antiparallel peptide arrangements because of the interactions between N and C terminus of the neighboring antiparallel peptides would even enhances the pore stability of these structures. So with that, we studied a lot of peptides, and we understood that this amphimaticity and the tight packing of non-polar side chains are not enough to stabilize the aptameric or large barrel pore structures. But we had to introduce some stacking interactions on the hydrophobic sides. But at the same time, we understood that these aromatic residues need to be at specific locations in the sequence for the formation of intermolecular stacking, because the intramolecular stacking could hamper the pore stability. And thus we optimized and we found the optimal positions for these aromatic residues on the hydrophobic patch, And these are, for example, on the first peptide-peptide interface, it is 13 and 13, and the second interface 15 and 15. And we ran the simulations where the stacking interactions on each interface were at least 75% stable, and the pore structure is also very stable in our simulation run. So similarly, on peptide hydrophilic patch, on the polar patch, we identified solvage interactions, intermolecular solvage interactions similarly, and we identified the most stable positions in the simulation run, the most stable solvage interactions on each and every each interface. I mean, on the first interface, on the second interface. We also identified the positions which led to intramolecular solvage interactions, which didn't stabilize the pore. So thus, when we have identified the most stable stacking and solvage interactions which stabilized the transmembrane pore structures, we formulated the design guidelines for new transmembrane pore-forming peptides, in which all the individual positions were identified, the roles, and these peptides were categorized into groups A, B, C, D, 4 groups, in which we combined the most stable interactions, which are stacking and the solvages. For example, in group A, there is 1 stacking and 2 solvage interactions per interface. And for each group of peptides, we proposed number of sequence patterns. For example, here I am showing for the group A, and in this sequence pattern, each and every position in the peptide can be modified with a number of amino acids. I am giving an example. For example, the position B is basic and can be modified with lysine and arginine, and A is acidic, which can be modified with aspartic acid and glutamic acid. So these basic and acidic positions interact with each other and form the most stable solvage interactions. Similarly, all the other positions can be modified, and this way, taking together the group A, B, C, D, we proposed altogether 52 sequence patterns which have the capacity to generate up to 10 to the power 15 sequences, which is a hypothetical number. These peptide sequences is capable of forming a transmembrane pore, where we know the role of each and every residues in the sequence. So now that all this information were from the design guidelines, were from simulations, so we wanted to validate them in experiments. So first we wanted to validate the pore-forming ability, and then we performed the fluorescent dye leakage assay. Here we made the lipid vesicles, which are trapped with the self-quenching dye inside fluorescent dye, and by peptide addition and the pore formation, the dye starts to leak, and there is an increased fluorescent signal, which is kind of similar to the ion channels. And this increased fluorescent signal indicates that there is a leakage. So similarly, we have studied 40 design peptides from the design guidelines, and here is a representative peptide. And we can see that at a concentration as low as 0.2 for the peptide concentration, there is already 90% leakage. But this is an indirect information of our validation of the pore formation by these peptides. But we wanted to directly see the pore structure, so that we performed the real-time electric force microscopy experiments. Here we prepared the supported lipid barriers, which is made of the lipids and which is completely flat before the peptide addition. But after peptide addition at 1 micromolar concentration, you can see that there are holes on the pores. And a zoomed image of the supported lipid barrier shows the stable and long-lived, because it was stable for at least 4 hours in our experiments. These nanopores, which are kind of 5 nanometer in diameter. So this was one of the representative pore-forming peptides. So now that we have satisfied with the validation, we wanted to demonstrate a practical application of this kind of peptides, these pore-forming peptides, and we chose to demonstrate the antibiotic applications, keeping in mind the increasing rate of development of bacterial resistance against the conventional antibiotics. So now the antibiotic peptides have a tendency or have characteristics which is cationic nature of these peptides, because this cationic character could attract these antibacterial peptides towards a negatively charged bacterial membrane over the zwitterionic human membrane. So we prepared the peptides by keeping the positively charged lysines at each ends, and that is how this P1 peptide killed antibiotic-resistant escape bacteria at micromolar concentrations, while showing negligible toxicity against the human cells. Here, this particular peptide showed the most promising activity against Acinetobacter baumannii and against E. coli, the resistant strains at 1 micromolar concentration. At the same time, they showed negligible toxicity against human red blood cells and human dermal fibroblast cells, where the 50% inhibitory concentration were more than 50 micromolar and nearly 100 micromolar. So you can see the therapeutic index, which is 1 by 100 here and 1 by more than 50 here. So that is how we demonstrated the antibiotic application of this peptide, which gives a starting point for the development of next generation antibiotics for these antibiotic-resistant bacterial strains. And at the same time, it also implies that these peptide pores can be used for the biotechnological applications like single molecule sensing and sequencing. With that, I would like to thank all my group members, especially my supervisor, Dr. Robert Baha, for his continuous support and motivation to this work. And thank you, SMPS3, for giving me this opportunity to talk about my research. And thank you, everyone, for your kind attention. Questions?
Thanks for my stuff. Interested in your design principles. Do you think you can apply the same principles or make new principles for beta barrels?
For the beta barrels? So these are specially designed for example, these are designed for the alpha helix, and beta barrel peptides have a tendency to have the hydrophobic and hydrophilic residues one after each other. So we can do that, but we haven't worked on the beta barrels. But people also have worked on beta barrel pores. For example, David Baker have worked on beta barrel transmembrane barrel pores recently.
Yeah, nice talk. Actually, two questions. So one is, do you know what's your pore insertion efficiency? Because you have the way your parts of your protein can also stick just to membrane. How have you actually quantified how many of them are on the membrane and how many of them are inside the membrane?
Okay, so we performed also the self-assembly process of these peptides, and in our simulations we have found that mostly the peptides wants to be in the, so it started from the surface adsorbed state in the simulations, and you can see that most of the peptides want to be in the inside the pore because they have this antibiotic character, and at the same time they are designed with the stacking and the solvage interactions which want them to be compacted with each other or aggregated, and that kind of aggregation at the same time antibioticity could be allowed only in the transmembrane region. So most of the peptide wants to be in the transmembrane region, but at the same time you can see that there are some peptides on the surface bound state. And it is also reported that this kind of peptides can be in the intermediate state. For example, some in the transmembrane, some in the surface bound. So.
Thanks. And the second question would be, actually, you showed in the last slide with antimicrobial activity, what's in the Tobacco baumannii. So as far as I know, because we also work with it, is that it has a strong sugar coating. Sorry. So Actinobacter baumannii itself is a problem because it has a strong sugar coat around it. Actually, nothing can go through. Do you know which variant of baumannii are used?
Which
variant?
Which barrier?
Variant.
Oh, which variant? For that you have to come to my poster because I do not remember, but it is reported in the poster.
Okay.
The which particular variant is there? I do not remember the number.
I have a question regarding your first slide, because you mentioned that these pores could be used to differentiate cancerous cells versus the non-cancerous. So I was just wondering what kind of design principles can be used to make the nanopores which can make it selective only for cancerous cells and not affecting the non-cancerous cells?
Okay, so similar to the antimicrobial activity of these peptides, which is the selectiveness is coming from the negative charge of the outer leaflet of the bacterial cells, and which is mostly negative, and at the same time the human cells, for the human cells, the outer leaflet is mostly zwitterionic, which is neutral. So the same sort of thing is also present in the cancer cells, which are, the outer leaflet is mostly composed of UPS lipids.
But is it the same for the non-cancerous as well? Like how would
So on the non-cancer cells, I mean, no, on the non-cancer cells, I guess, okay, so there could be some other specificity which could be defining it. But I haven't worked on the cancer cells, to be honest, so that's fine. Yeah. But the specificity usually comes from the negative charge and the exudative character.
Yeah, I mean, I completely understand its application for antimicrobial and antibiotic, but I was just wondering like how we expand it for cancerous cells because they are different, right? I mean, the normal cell and cancerous cells, very similar composition of the membrane. That's why I was wondering, have you thought of any design principles?
But the local cancer environment is often acidic, so you could design that to take advantage of that.
Okay.
Right? Questions? Yeah.
Very nice talk, thank you. I was wondering if you can apply these design principles you use to bacterial, but can you go into yeast as well? I know there's like a preventive
Sorry,
yeast cells.
I have difficulty with this.
Yeast?
Yeast cells?
Yes.
Okay.
Apply some of these design principles to target yeast membrane rather than just the bacterial.
It is possible. It is possible, of course, yeah. Because some of the collaborators from our institute are also working on that kind of things. Yeah.
Thank you.
All right. Thank you very much.